So good morning, everyone. Like Noé told, there's been quite a few people during this conference who've been talking about EpiGBS. And today I'm finally going to explain how it works and how well it works. But first I want to give a quick shout out to Fleur. She's not here, but she's been very instrumental at getting everything organized surrounding this pipeline and everything being coordinated. So let's see which one is the one that changes. This one. So in 2016, the first version of EpiGBS was published. It was made by Thomas van Gerp, who was a PhD student at NIOO at the time, and Niels Wagemaker. And it's a reference-free reduced representation bisulfite sequencing method. That means that we can have DNA methylation information on a nucleotide level on a small, untargeted but replicable subset of the genome without needing a reference genome. So this sounds all fantastic, and I think it is. But when I started in working with this, we found that all the algorithms in this pipeline were custom-made. It was very challenging to install and very hard to use, leading to lots of moments where my computer said no to the data coming or data coming out. So together with Fleur and Samar and Morgan, who are part of the EpiDiverse project, we decided to try and see if we can improve this method. And then we had some specific goals. We wanted to make the pipeline more easy to use more proven methods, to make the entire pipeline more reproducible, and we wanted to include a reference branch to ensure you can also use this pipeline for model species instead of just for non-model species. So in 2020, we published a preprint, but since then we've further developed the pipeline a bit more because we added another star to our already star-studded team in the form of Adam Nunn, and he helped a lot with creating and helping to improve our SNP calling capabilities in the pipeline and also the benchmarking for the SNP calling. So this paper is going to be submitted today for the first time, but the pipeline is already available at the GitHub. So I will now quickly go through how EpiGBS works. I have to do this quite quick because there's not enough time to go in very much detail. But if you have specific questions, you can always ask after the presentation. So you start with your DNA on a plate. This is then digested with restrictions, enzymes, and barcode adapters are added. So this looks something like this. So you have your entire genome, and it's cut using restriction enzymes at certain positions. This fragment is then attached to the barcode, so we can sequence it on the Illumina system. And there's a unique molecular identifier, an individual barcode, and a control nucleotide. And these will all come back when I explain how the pipeline works. After this, there's a cleanup step. There's nick repair to make sure that the restriction sites are working, and then it's bisulfite converted. So this means that all of the unmethylated cytosines are converted to Ts. And then you go to, there's another cleanup step, and then you send it out for sequencing. So I haven't done much lab work on EpiGBS, but the new paper will have a very extensive laboratory user manual. So for any more questions on that, I would like to refer to that because I'm not that much experienced with it. But I am experienced in the part where we analyze the data. So what happens after you get your results back from sequencing. So I'm first going to explain a bit about the things that we use to make the new EpiGBS more user-friendly. So first of all, we have Bioinformatics. And Bioinformatics is a program which allows for very easy installation of programs, and it makes sure that everyone who uses your pipeline actually gets the same version. So in this way, you won't have very different versions between all of the people who use it. So if you now download the EpiGBS pipeline, you will get the exact same pipeline that we used for benchmarking, and so results are expected to be more consistent. And next to this, we use SnakeMake, which is a workflow manager. So this ensures that you can easily start the pipeline by filling in a nice form with certain parameters you want, and it will then run the entire pipeline without you having to worry about, oh, is this part of this pipeline finished yet? I need to turn it on again. And yeah, so it's way easier. And then we have MultiQC, which is a very flexible program which you can use to plot all sorts of data that is related to the performance of the pipeline. And we also include some specific things that are specific for EpiGBS. So you can find things like bisulfite conversion rates in there as well. So this is how the pipeline now looks, and I will quickly go through this. So you start with demultiplexing. So if you remember, these are the reads that you get after the lab work. So there's a universal, we do the demultiplexing using stacks, and there's a PCR duplicate removal based on a unique molecular identifier. So these are 3 bases that are randomly added to each adapter, and based if they are replicated, it's probably a PCR duplicate. Then there's the barcode, which is an identifier for each individual. And then there is a control nucleotide which allows us to differentiate between Watson and Crick and allows us to create the DeNova genome. So for the DeNova creation, first the Watson and Cricks R1s and R2s are combined. Then we make all of these reads binary. They are deduplicated. And then from these deduplicated binary reads, we reconstruct the reference based on the information that we have on the Watson and Crick reads. And afterwards, these reads that we are left are again clustered and then based on identity. So you set a certain threshold, and clusters that are more similar than that threshold are then grouped together. Afterwards, we do mapping and methylation calling, and for this we use Bismarck, which is widely used, is well maintained, and it really interfaces quite nicely with common analysis packages such as DSS and MethylKit, and also all sorts of bisulfite sensitive SNP callers. So this was a quick overview of how the pipeline worked. So now you're probably all very interested in how well this pipeline works. And for this, I really need to thank Morgan van Antro, because she provided us with 44 individuals of 6 accessions of Arabidopsis thaliana, and not only grew them in the greenhouse, but also did all of the lab work. And with this data set, we decided to see how well the pipeline performed. And there's 3 different things that we're really interested in. The first one is the DeNova creation. So how well do we create a DeNova reference compared to the Arabidopsis reference? How well does methylation calling perform? And then we compared it to the 2001 epigenomes. And how well does the SNP calling perform? And then we also compared it to the 2001 genomes. So the DeNova that we created, it's with a specific parameter combination, but yeah, that's maybe something for another day, how the parameters cause the differences in how the DeNova performs. But in the DeNova, with the most optimal parameter combination that we could find, we found 1 million total cytosine sites, of which 928,000 mapped once on the Arabidopsis genome. So there might be some sites that were multi-mapping, but these were also excluded. But if you would have a reference, it might actually they might actually do map on it. And this is about 3% of the genome that was covered. So in this way, like, the DeNova performs quite well, I feel. When we look at the methylation calling, this is a quite complicated graph, but the x-axis is the fraction of methylation in the DeNova branch of EpiGBS, while the y-axis is the methylation percentage in the 1001 epigenomes for all the different sites that were covered. And you see here that we can very accurately, with an R-squared of about higher than 0.9, we can accurately, the sites that we find the methylation sites are very similar in the CG and CHG context, but there's a very weak correlation CHH. However, as we've seen this last couple of days, CHH is very variable, and the Arabidopsis samples from the 1001 epigenomes in 2016 are not necessarily the exact same plants. Well, we know they're not the exact same plants grown in the exact same conditions as the plants that Morgan grew. So this is the DeNova, and the reference branch actually performs just as well as the DeNova branch in the methylation calling. We do find more sites, but this might also be due to the limitations of the DeNova creations. So then we'll go to the last part of the pipeline, the SNP calling. For this we use a masking method developed by Adam, and we do SNP calling with freebase. I'm not going to explain how the SNP calling works exactly, because Adam's going to have a talk after the coffee break, so I would recommend you to stay for that. Here we compared EpiGBS to the 1001 Genomes project, and we used RTG-VC-eval. And first I need to explain two or three terms before I can show the plot, because otherwise it's going to be quite complicated. The first one is precision, and precision is the number of true SNPs discovered divided by the total number of SNPs discovered. So in this way, this is a measure of how many of the SNPs that you found are actually real SNPs. And then there's sensitivity, which is the number of true SNPs discovered divided by the number of SNPs in the 1001 genomes. So this is a measure of, well, how well does the pipeline perform to find SNPs? How many of the SNPs does the pipeline find? And then there's the quality, which is an output of freebase, and this is a confidence that freebase outputs to say how confident it is in the fact that a SNP is actually a SNP. So these are the sort of plots that you get out of this. So this is the reference branch, and here you find. So here we have two lines, and these lines start at basically all of the SNPs that freebase have, including low quality ones. So if you remove the low quality SNPs, you get less SNPs, and your sensitivity goes down, which makes sense. But since the low quality SNPs are not as likely to be SNPs, your precision goes up, and that means so that the SNPs that you find are more reliable. Then we have the blue line and the black line, which is a sort of a filter to exclude sites that EpiGBS cannot really call due to the RRBS nature of it. So these tools were developed for whole genome, where you sequencing whole genome. Yeah, basically whole genome sequencing, but reduced representation and the combination of bisulfite, there are some sites that it can not really call. But so if we filter those sites, we see that about 80% of the SNPs that should be present are found using EpiGBS, and it calls them with an accuracy of about 96%. So it's very accurate, and it finds most of the SNPs that it should be able to call, and it does that with high accuracy. So with that, I'm going to go to the conclusion. So we have now this new EpiGBS2 lab protocol and pipeline, and they're very well documented. They're easy to install, easy to use, and have good reproducibility. And benchmarking has shown good performance of both the SNP calling and the methylation calling. So before we go to the questions from you guys to me, I want to ask everyone who wants to use the EpiGBS a question, and that is try and contribute. And then I don't necessarily mean write code or anything, but I really, really would like you to, if you have any issues, instead of sending emails to me or Fleur, please ask them on GitHub. In this way, people can very easily Google their issues, and if the issues are solved, they will still show up on Google, and people can just find them on the GitHub instead of having to have multiple mail conversations with all different people who try different things. But in this way, everyone who uses the pipeline can together help to improve it and help people find solutions for issues they encounter. So with that, I want to thank you, and I want to thank a lot of people who've worked on this project and helped testing and reporting, but in particular Fleur and Morgan and everyone at NIOO who's helped with the lab work, and my supervisors. And well, there's a lot of people involved, but I want to thank everyone. So with that, now you can ask me questions.
Thank you, Maarten. We have some questions here from Christine.
Wonderful. Thank you so much for that. I really appreciate it. And I also apologize for being one of those people that has sent you 1,000 emails, but you've always been very gracious, and I appreciate that. So I have 1,000 questions for you, but what I'm really interested in is that you reported only a 3% coverage of Arabidopsis, which I find surprising. Do you think that's just because of the depth of how much sequencing effort you put into it, or you needed more, just throw more data at it, or is that?
Yeah, there can be quite a few things that influence that. But first of all, the DeNova that we created was quite stringent in filtering. So the deduplication was at about 100 reads, and then you have afterwards you get the clustering identity. So I think there's quite a lot of wiggle room in how extensive you make your DeNova or how small you make your DeNova. And yeah, so that is quite a big influence on how much you cover. So it's a bit of a trade-off between how certain you are that these are actually reads from Arabidopsis that are constructed in the correct way versus how much of the genome you try to cover.
Right. And when you said 3%, did you mean 3% was sort of the average for the 6 different, or you had 6 different or 5 different?
No, yeah, so we created the DeNova from all 6 of the different lines.
Okay.
So, yeah, but there is. I haven't quite figured out myself yet what. Yeah, it's quite hard to figure out what a good DeNova is. Like with Arabidopsis, it's quite easy. It's relatively easy to figure out because you can compare it with the reference genome. However, if you use non-model species, you can't really compare it to anything, and then you just need to get some sort of a feeling to find out when the DeNova is contains all the things. That you want it to contain. So. Yeah, it's something I'm working on as well to try and figure out what are the most optimal parameters for different species and why that's the case.
Yeah, great.
Okay, I think there's a question
there. Yeah. What do you think are the most important indicators that your DeNova assembly has gone well? Or are there any pointers which tell you that the DeNova assembly did not go well? Well, so I can go into this a bit more. So these are I've created this option that I'm still developing, which shows how the different parameters actually influence the DeNova creation. And you can see, so depending on how strict you cluster or how many reads you need for samples to pass deduplication, you can see it influences all sorts of things like the total number of clusters, the percentage of the clusters that map on the reference, the number of clusters that reference. So these are, you can't get those from non-model species, but you can get like the mapping percentage, the average depth, and that kind of parameters. And I think these are quite, quite important indicators like average depth and the number of clusters, but it's quite hard to, you need to see how the parameters affect those different indicators. So I'm not completely sure on what is the exact way to do this.
Okay, I think we had a question over there.
Well, thank you very much, Maarten. I really appreciate all the work that and all the effort that you've put into developing this pipeline and making it more user-friendly, especially for non-bioinformaticians. And I have a question regarding the CHH methylation estimates that you're getting, which don't seem to be correlate well with the estimates that have been before. And I wonder whether all of us that are working with EpiGBS, and I'm finding patterns in the CHH context, like it is more responsive than to the environment, should be worried about that?
I don't think we should be worried about that. That it's because if it correctly calls CG and CHG, I don't think there's any reason to assume that suddenly for CHH the bisulfite conversion doesn't work anymore. So I don't think that's a bioinformatician. I think that's a biological signal and not necessarily a bioinformatics issue. And I think that's mostly due to the distance between the 2001 Epigenomes project and the plants that were grown at NIOO. I'm not sure. I haven't seen much work from other people who've done this with epigenetics, so I'm not quite sure how this compares to if this is something that happens all the time with this context. So.
Okay, thank you.
Okay, do we have more questions? Or?
No more questions.
Okay.
The screen.
Then thank you again, Maarten.
Yes.